All right, let's get started. Hey, everybody, welcome. We've got our sunflower team. We've got Kevin and Bob speaking today. And then we have a couple folks from the Land Institute. We have Pheonah Nabukalu and Brandon Schlautman, who's going to be talking about perennial sorghum and Sanfoin. And I asked how to pronounce that, and that's one of the ways to pronounce it. So thanks, everybody. Excited to have you here. As always, I want to start off with an acknowledgement of Forever Green's funders, the Environment and Natural Resources Trust Fund, the Minnesota Department of Agriculture, Clean Water, Land and Legacy Amendment, Builders Vision, and the McKnight Foundation, among others. We're going to move on to announcements. Mitch, any announcements?
Really briefly, I want to make everybody who's not here in person feel bad because there are hazelnut macaroons in the corner. And so if you're not in the habit, I mean, obviously if you live out of state or whatever, but if you're here and you're not in the habit of coming in person, Claire's upping the bar on that. And I just had to specify that willingness to bake on a regular basis was not in the job description, but I'm not sad that this turns out to be one of Claire's many fine characteristics as a Forever Green person. So thank you again, Claire. And then the last thing, my only other announcement is I hope everybody saw, well, first of all, I hope everyone is on the list to receive the now weekly Forever Green newsletter. So we are very fortunate. Also speaking of other newish staff, Ann Kirby and Cora Jensen put together our weekly newsletter. And I think it's doing a great job of keeping everybody informed of what's going on. If you're not on that list, you can reach out to Claire or me or Ann or Cora. So this was shared over the newsletter, but I just want to highlight it again. Very cool scientific milestone. Pennycress was on the cover of Nature Plants, this latest issue of Nature Plants. And the paper was led by John Sedbrook and his team at Illinois State. And for those of you who've been around, you remember that John and David Marks are longtime collaborators in the domestication of pennycress. John with a CRISPR-based approach, David with the EMS mutagenesis approach. David's a co-author, and Ratan Chopra, who's a Forever Green alum and now with Covercress, is also a co-author. This paper is really highlighting the John side of the story. David's working to tell his side of the story, and another paper that's in the works. But it's just really exciting, really validating, I would argue, to the fact that this work is at the cutting edge of science. So really cool to see, and congrats to John and all of his collaborators.
Okay, I wanted to update you on where I left the perennial sunflower project. This is a list of the people that have contributed to this project over the years, and starting with Don Weiss to Yaniv and Brent Halke and Mikey Cantor. And I'm the common thread that runs through all of this, and I'm not the smartest one by any means. I'm just the one that kind of has lived through all of these phases. So as an update of where we are now, well, this is where we started with the idea of developing a perennial oilseed sunflower. And there were a number of groups that were working on this, crossing annual sunflower to various wild species. And we had the most luck crossing Helianthus tuberosus, is commonly known through some artichoke, with a diploid annual sunflower. There is some problem with hybridizing these, not the least of which is the chromosome number. But we were able to get the diploid annual species and the hexaploid tetraploid species produced a hybrid that the hexaploid perennial produced a tetraploid hybrid. And this was fairly stable. The seed production was very low, but whether that was due to genetic difficulties or the fact that wild type generally is not a very good outcrosser either. So it could have been either of those two things. Eventually, we tried recurrent inbreeding with the idea that the self-fertile trait should be in here somewhere, as it, since it was crossed with the annual. So just through brute force recurrent selection, we got only a few seeds, self those. And eventually we were able to find some individuals that were able to self-fertilize and set seed. And this was a greenhouse shot of probably the first one that was one of the most exciting. This is like I think this is about 3 generations of selfing that we found this one. And generally through selfing of these lines, we were getting like 1 or 2 seeds. And at this stage, this S3, I got 206 seeds. I thought we were on our way at that point. See, one of the concerns we had with this was that once we started getting self seed production off of these things, we were maybe moving more toward the annual traits. And the concern was that we would lose the perenniality, but that didn't appear to be the case. This is that same plant. Well, this is a precursor to that plant, but we seem to be maintaining the perennial characteristics as well as getting the seed production, which was a big step in where we wanted to get to. Anyway, moving forward, we have a number of lines where we've used this selfing protocol over 4 to 6 generations, and I wanted to evaluate the phenotypes and the productivity and the phenotype of these inbred lines in St. Paul. And that's what I've been doing over the last couple of years. And these are only small plots. They're 2 rows and only 5 feet long. But the idea of this was to actually measure the yield compared to what would be a real crop sunflower variety, and also to see how well these would actually overwinter and produce in a second season. This is a little bit out of sequence, but our selection procedure for this was trying to get something that looked exactly like annual sunflower, which means a single head phenotype, large single head phenotype. And so I have a number of inbreds that match this phenotype pretty well. The other phenotype would be the multi-head phenotype, which I kind of selected against over much of the time, but when we started doing yield calculations, we found that these actually tend to yield better than the single heads. So I have these in the stable of inbreds as well. These are the yield results over the last 2 years that I've done this. These would be the best inbreds, and these are all from different inbred lines. The first yield column, this is in kilograms of seed per hectare. So last year some of these were quite productive, but at the very bottom there's an annual check. This is an inbred annual, so this yields slightly less than would be a hybrid sunflower variety, but the inbred annual sunflower check was yielding a little over almost 2,200 kilograms per hectare. And the best inbred line that I had last year gave me 2,054, and that was one of the multi-head types. So that very best one was actually matching the yield of an inbred, which is slightly lower than would be a crop variety. But
how is the maturity of the multi-head ones, and how much do birds kind of get in there?
Well, the birds are always an issue, and they do. In fact, this year, well, I've been harvesting these things probably a little sooner than you would in the field. So that's one thing. I harvest them early and take them and dry them down, but birds like them a lot. So yeah.
So is that like a disadvantage of the multi-head if they mature
Well,
at different, or do they all mature at the same time?
They mature at different times, but it's not that spaced out.
Okay.
It's almost at the same time, so that's not a huge issue.
Okay.
One thing that annual sunflowers do, you'll notice, is that they will tip upside down, and supposedly that helps prevent birds from eating them. And these don't do that generally, so that it makes a really nice perch for a bird to land and just feed. So unfortunately, that's That's one of the problems. But anyway, this of the single head types, the best one was closer to well, you see over a couple columns, there's flowers per shoots, and you know, the closer you get to one, that's the single headed type. So the second highest yielder was more of a multi-head type, although not quite as multi-headed, and that was about 1,500 pounds. And then the best single-headed type was about 1,000 or kilograms per hectare. Next major thing of this is the winter survival. And for the most part, well, there were a couple that did quite well. You see 7 out of 10 survival and 8 out of 10 survival for some of those, and some were really bad. Some didn't pull through at all. Some only had 2. So it was a bad overwintering winter too. So I'd like to see what happens through this winter, because as cold as it's been, we've still had a lot of snow, and that's kind of the key thing, is whether you have snow cover or not. So I hope it does better this season. But I mean, there's overwintering there. It needs to be a little bit better. The yield could certainly be better, so it's got a ways to go, but I think we're on the right track.
Have you noticed, like birds have a harder time landing on the smaller multi-head ones?
Yeah, they don't, well, I'm sure they do. I've seen them on those as well.
Okay.
And I'll show you some pictures of what these look like.
Having on the overwintering, just with this table,
Yeah.
like the bottom hybrid shows 0 overwintering.
Yeah, and that's the point I should bring up, because, you know, I have yields listed for this year as well. And the reason I have yields for something that didn't overwinter at all, and this is a caveat to the second year yield, is that wherever a plant died, I backfilled it with a clone from the greenhouse. So that's cheating. This isn't a valid second year result. That, I mean, some of them did overwinter, and I'm hoping that this winter maybe we'll have a little bit more coming through. But
Kevin, was there any ice sheeting? Because I know, like, again, like 10 years ago, that was always a big problem if you had any ice sheeting versus no snow. Like, I mean, like, and I know that was like a bigger problem in K rather than X, the K fields rather than X, the X fields.
Yeah, I don't think that was the problem. There just wasn't any snow. But we did have, over the last few years, we've had periods in January that have been kind of dry. And if you don't have any snow to begin with, and then it will get cold again, and there's absolutely nothing to protect the tubers. I think in this case, I think that's what happened. I don't think it was ice sheeting.
It was a really hard winter for overwintering. We had, not just here on campus, but across the state, a lot of cereal rye that died, which is very, very unusual. So it's just that lack of snow and cold temperature combination.
Right.
I just to emphasize that when I look at that table, what I see first and foremost is 7 out of 10 and 8 out of 10 is really high considering what the conditions were like that winter.
Well, yeah, I would agree.
And I think that in an ordinary winter, those numbers would all be much better.
Yeah, and well, in those, if we take those as truly overwintered plants, I do see the yields dropped off still quite a bit. I don't know if this is something that we've seen so many times in perennials or not, where segment third year drops off. I hope not. Part of it was quite a bit of bird issues. I didn't get on top of it as early as I did previous season. So the other variable things with these phenotypes would be like flower size. The largest flower that I had this year was I think 7 centimeters, and this compares to an annual. What's an annual at about 20, maybe. So they're getting larger, but I mean, I don't think they will ever quite match what an annual sunflower head looks like. You know, even when I say single heads, this is a single head per shoot. So if you have a plant, the plant may have 8 or 10 shoots. So one plant is probably going to have 10 flowers, even if I'm calling it a single head, just because I'm counting the number of seeds per shoot. The multi-heads will have a shoot with multiple heads coming off of it.
Do they shatter at all without birds?
Without birds, I don't know. I've seen shattering, but I think it's birds that are doing it. And the seed weight per 100 seeds in grams, this, you know, if you look at the annual, this is, it's like a little over 6 grams per 100 seeds. And this is a third to a half of that. So, and HA89 isn't a super big seed either. It's and well, in fact, maybe I had this one. Actually, I had a seed comparison on here with to the far right of that. You see the same seeds going down on the right column. Those are all from there, and on the left are different either annual or yeah, these are different annuals. Sunflower cultivars. The top large one is Paradovic, and HA89 is like the third one from the bottom. So it looks about the same size, but it's also the annuals are also plumper too. And even though it looks like it's almost the same size, the actual nutlet in there is a little bit flatter. So there's lots of improvement that could be done on this still. Okay, well, these are just some pictures of what I've been talking about here, and I put the table data up here so we can kind of keep track of that. But this is the annual HA89 inbred line. And again, this was 2024. That was 2158 kilograms. This one is an S5 inbred. These are all starting from different lines. And this one in 2024 was 870, and this one was a 7 out of 10 survivor. So this one was pretty good in that regard. It's a 5th generation self. Again, the heads are a little larger. The seed yield is, again, about 800 plus. This one didn't survive at all. I do like the looks of it for various reasons. I like just the stature of it in general. This is a derivative of the one I started with. So this is an S5 of that line that I introduced you to at the very beginning. And this one in 2024 was 1116 kilograms, and this overwintering happened at about 50%, which is not too bad. And I like the stature of this one, and the single head type is something I like, although yield wise, maybe it's not that smart.
When you say S5, does that mean
Self,
Okay.
5th generation self.
So, okay. All right.
Yeah, this one, again, this is a good yielder. Some of my text got clipped off because I'm not very good at this, but this was a good yielder, 1500. The winter survival was quite low. Only 2 of them came through the winter. Okay, here's an example of one of the multi-headed ones, what they look like in the field. And this was the best yielder last year. See the number of heads, and they're all fairly uniform in maturity too. That's kind of what you get. But I mean, one problem with perennials in general is that the flowers are mature here, and you can see there's still a lot of green material. So, you know, for mechanical harvesting, something would have to be done. It would have to be desiccated in some way in order to harvest this. Otherwise, the seed will be full of wet material.
There's a new desicant out there called pelargonic acid that seems to be pretty good.
Really?
Yeah,
You use that for your
I read about it,
okay.
but it's a fatty acid, but it gets in the cell of the plant cells, and it just acidifies the cell wall, and yeah, it's a pretty good desiccant, it looks like.
So,
Kevin, do you think
Yeah.
you could wait for these plants to dry down and then just have this later, or do these stay pretty green until
Well,
like October?
they'll stay green.
Okay,
Well, they'll get drier,
yeah.
but
definitely.
yeah,
Okay.
but they'll be pretty tacky right until the
Oh,
time
yeah.
it freezes. I mean, if you wait until
Like-
it freezes, yeah,
Yeah,
but that's gonna be pretty late in some cases.
yeah.
And the birds will have ample chance to eat everything that's there too, so. So there are some issues with that, but, and this one overwintered pretty well too, so. Okay, and this is, I have just a couple more slides. These are taking a different tack. I still have a number of lines that produce almost no seeds, but they're, phenotypically, they're very nice plants. They just don't produce seed. This is one example of one of those plants that I crossed one of the more fertile plants to. And then from that point, I did a number of cells. So this is one of the sort of semi-success stories of that. And this is, the yield of this isn't tremendously high. It's only 560 kilograms last year. The winter survival was so-so. It's 4 out of 10, but I really like the stature and the phenotype of this plant. So it's one of the things I'd like to improve upon. I had one more here. Oh, this one I kind of missed, but this is another one. Seems to mature a bit later. Again, this is one that comes from a line that had very poor seed production, and I crossed one of the better fertility lines to this one, followed by selfing. And what I like about this is, the seed yield was about 950 last year, which is okay. The winter survival was 5 out of 10. I like the single-headedness of this, and I've been pointing out the heights of this, but this is kind of a shortish plant, which is a nice thing too. It seems to mature a little later, but I like the sort of semi-poor stature of the plant. So this is one that, and it's kind of pretty too. So I kind of like this one. This is kind of disjointed, sort of stream of conscience thing for me. Okay, to tie it up, the future work, I mean, using the inbreds that we've developed, the next stage would be to intercross these inbreds and see if we can get some sort of heterotic improvements in yield and maybe overwintering ability and that sort of thing. The other thing that Bob Stupar's lab will be picking up would be the DNA sequencing of some of the selected lineages that we've looked at here. And this is not my strong area. So if I say something stupid, just discount it. But I think, this would allow, us to identify sequences that would be associated with some desirable or undesirable traits. And maybe, if you're starting from scratch, maybe with some brand new annuals and perennials, you do some intercross and look for some of those markers perhaps for something that's worked already, and then you could just direct your breeding program in a little more efficient way. And I'd like to know what the relative contribution of the annulus and tuberosus genes are as we go through these generations of selfing. If one, maybe the tuberosus is diminishing and the annulus is strengthening, and what the ramifications of that are, I don't know, but it'd be interesting to know.
Beside the yield, what else treats is yield in number one treat? What's the number two treat? Must have
Well, the ability to overwinter would be my number 2 trait.
winter hardiness.
Winter hardiness would be my number 2, and also there are a number of other ones too. Seed size would, obviously, larger seed size would be a great benefit. There are things that I haven't even considered yet, like drying down, like early maturity and the ability of the plant to dry down on its own. That would be a nice thing.
So I'm gonna try to explain this nomenclature a little bit. So a lot of these materials have been first hybridized and intermated and self. That's basically what Kevin's been talking about. And most of the traits that we really care about, I think, are pretty good with just the hybridization and intermating. Like, things are quite perennial, and they're, they survive winter, and even some of the architecture traits are really good. The problem is its fertility is low. And that's why Kevin came up with the idea of self, is because by selfing, if fertility is low because of the chromosomal ability to go through meiosis properly, which makes sense because they're two different species that are coming together. The idea is if you self them enough generations, you'll eventually create inbred lines that at least do meiosis properly. So one of the questions we have here is, can we select for fertility and still maintain those perenniality traits? Or are we going to lose? What are we losing from that? So this is kind of, I guess, the fundamental question that we're really addressing. What has Kevin created? We don't really, genetically speaking, we don't really know. So that's really the question that I think we're most interested in. So basically, Kevin talked about this. What is the contribution of the annual and the perennial genomes in these self lines, these best self lines? And the other questions we have are like, is there recombination occurring between the subgenomes from the perennial and the annual parent? We don't know. And then ultimately, what would make a better product if we continue to select within the best inbred lineages, or if we take the inbred lineages and then try to make F1 hybrids with them, kind of the way we do with annuus, like the annual perennial, or the annual sunflower is a hybrid sunflower. So those are things that we're hoping to address with this part of the question. So this is just a diagram of how this actually works at the chromosomal level. I'll show you the simplest model of what we have. So on the left, you see sunflower. Kevin mentioned that's got 34 total chromosomes. I'm just showing 8 of them for simplicity. And then on the left, Jerusalem artichoke has 102 chromosomes. Pointer's lost. But anyway, and so you can kind of see I've drawn them as subgenomes. So this group of 4 pairs is analogous to that group of 4 pairs there. And that's the second subgenome, and that's the third subgenome. And we think it behaves in a way where pairing is, we actually don't know exactly. I don't think we know how exactly Jerusalem artichoke pairs. But the big point is that we think that the genome on the left, let me see if I can, here, maybe this will do it. Laser pointer. Yeah, there we go. So the genome on the left, we think probably pairs favorably with one of the subgenomes, just based on what we know about the evolution of Jerusalem artichoke. And we hope, if that's the case, that we can get an F1 that looks kind of like this. This is how the F1 pairs. So basically, this big chromosome here is inherited in the F1, and then that chromosome here is inherited in the F1, and then they would properly pair during meiosis. But maybe they don't, right? Maybe that's what kind of hurts the fertility of these plants. So you can get some fertility, but maybe not great fertility. And then over here, what we have is we're inheriting this big chromosome and one of these big chromosomes, and we're hoping that those sort of pair, because those subgenomes are, we think, more similar to one another. So it kind of we hope this kind of behaves like a diploid. And then what we then do is we cross, this is what they call the intermating step. We take this F1 and we cross it to another F1, for instance. And that creates what we call the intermated 1 generation. So now we have something funny happening, right? Mendelian genetics predicts that these will, let's just say there's no recombination. These two are going to independently separate from one another in segregation. So we might inherit this yellow chromosome, and we might actually inherit this yellow chromosome from over here too. Or we might get both maroon ones. I'm just showing you one possible outcome here. So in this possible outcome, I've said we get the maroon one as homozygous here, we get the yellow one as homozygous here, and then the other two are heterozygous. Just one possible outcome. For the sake of this discussion, it doesn't really matter what happens exactly here. Then we intermate that with, say, another one of these intermated F1s that has a different chromosome composition. And then now we would have an intermated 2 generation here. And we continue to do this, basically just take, what Kevin does, he takes the good and he crosses them together and sees if he can make something better. And you do that for at least 4 generations, and you end up with something that maybe, this is where the question becomes. So after several generations of intermating, we continue to see phenotypic improvement while maintaining perenniality. That's the whole point. That's the breeding piece. And then the question that we're asking the genomic side is, does this mean we're effectively selecting for enrichment of the domesticated chromosomes? In other words, are we, through rounds of selection, purging the maroon out of here, and that's why we're getting better fertility? And maybe that's why we're getting single-headedness or moving towards fewer heads. In other words, can we select for the domesticated genome through this process favorably? And then we get to selfing. So now, after 2 rounds of selfing, or 4 rounds of selfing, which I'm showing you here, now we can create something that maybe is inbred in true breeding and won't segregate for traits in the next generation with enhanced fertility, because now the chromosomes will pair properly. That's the idea. So after several generations of selfing, we can still see improved fertility while maintaining perenniality. And does this again mean that we're effectively selecting for that domesticated? So let me show you why this is important. Let's say this is our product. On the previous slide, where we started, was this. You can see we have very little yellow up there. But by the time we do this selection scheme, we could perhaps get like half of our genome back to domesticated sunflower. That's sort of the idea. Okay. Now, I have a couple of slides that are going to complicate this a little. So now we're going to bring in recombination. So it's actually probable that the subgenomes recombine. And so we're not doing this as cleanly as what I just showed you a moment ago. Instead, you're getting this this intermated 1 that's going to have sort of segments of chromosomes from each parent. But we can still select for the best types, which presumably will enrich our annual genome presence. So again, we do an intermated 2, and now we get this like puzzle, right? Pieces coming together. And as we select, so now we're selecting for not chromosomes, but segments, or parts of chromosomes. And over time, we can self those out, and we might get something like this by the intermated 4S4. We have some, because we're again selecting for annual and fertile traits, maybe we're selecting primarily, we think, for the domesticated genome, but bringing in the perenniality traits, presumably from the other subgenomes.
Bob, quick question online. What does enrichment mean in this context?
Enrichment means we're getting more of the yellow genome as we select over generation. So we're enriching for the yellow genome, which in this case is the domesticated parent genome. So that's what I was saying a moment ago. We go from like this, which is this genome right here is 25% yellow. By the end of this selection, we're coming up on 50% yellow. And maybe perhaps that's why we're starting this. And this doesn't even account for the possibility that the subgenomes interact, which if they do, makes it possible to select for even more yellow over time, because you get more novel recombination events across species, the subgenomes. So this is all theory. When I'm telling you, we don't know if any of this is happening because we haven't genotyped any of this stuff. But the idea now is that we can go into Kevin's pedigrees and start to genotype these lines. This is the sequencing part of the project. So what you see here is a pedigree that Kevin created where he crossed the 2 original Jerusalem artichoke and domesticated sunflower. And then here what you see is that intermated 4. So that's what the IM4 over here sits. I can barely see it, but that's what that says, IM4. And then he starts selfing, and this becomes a family, all derived from one IM4. This is what he calls D family. And so from this, he makes selections. So this is the S3 generation here. He's got a couple selections. And the S4 generation, he's got a couple selections. And the S5 generation, he's got a couple selections. These are all different clones now, tubers that can be grown over and over again. And then he's got some plants that he's also maintained that weren't selected, but are also perhaps of interest to see what the genomes are looking like. So the idea here is basically that we can take the good stuff and see what's been going on in the history of these chromosome rearrangements or recombinations or selections. So what we have to date, we met recently on this, is we've selected about 4 or 10 different IM4 parents from different families. So I just showed you the D family here. We've got about 10 of these that are all different They're all derived from different intermated pedigrees. And then we've got about 10 that we've selected from the D family that are down here, the selfed versions. So we have the IM4 parents, and we have the selfed versions that came out here. And then we also have inbreds from some of the other families as well. And the idea that we're trying to build here, this is where it gets kind of, it's already complex, but this is where it gets more complex. So Kevin has a pretty good idea what these phenotypes look like. And we could just say, let's hybridize these inbreds as best we can and just hope for the best. Or we can look at the genomes and say, we can actually predict that these are the two most distantly related inbreds. You can cross them together and maybe get a bigger heterotic effect. Or perhaps it's the opposite. Maybe we would crossing narrow because they maybe have higher fertility. We don't know. And that's kind of what we're trying to ask that question. So this is where it gets back to this question is, can we use this genotyping information to predict what crosses will be successful, will make new good parents, will make new good inbreds? And then ultimately, is an inbred or a hybrid path the one that we should be going down? Is there an inherent trade-off between vigor, which you get from hybridization, versus fertility, which you might get through self-pollination? So that's something that we're kind of thinking about. And can we use this information to predict the best and worst crosses and then eventually empirically test that prediction?
Do you think back crossing at some point to the annual would be helpful?
Back crossing to the annual. Oh, yeah. So that's a really good question. And I think the answer is it would be awesome if we could. The problem is a cross between the tetraploid and the diploid will make a triploid. And the triploid will then be probably zero fertility. If we could figure out a way, and I know Brent did this years ago, Brent Hulke, where he tried to double the diploid and then you make that as, that would be an ideal way to do it. It's just the attempts that, Mikey worked on that material a little bit when he started his PhD, and it turned out that I think he could make good triploids from them, but I don't think that any tetraploids.
Yeah, there were two problems. The triploids had almost zero fertility, but the bigger problem was none of them formed tubers.
They're not perennial, apparently.
And so they didn't form tubers. And with we did do the colchicine doubling, and the problem that we ran into with those is we weren't able to get stable tetraploids. So you could generate something that looked like a tetraploid parent. Then when you selfed it, you ended up with triploids or just diploids because there wasn't clean colchicine doubling. And that was also true. We got seed from Grin of the putative tetraploid lines that had been developed by the USDA cytogeneticist CC Jan, and they were no longer tetraploid by the time Grin gave them to us. So, yeah.
And that's the other thing I didn't really talk about. I feel like this was complicated enough, but the other thing that we talked about within the smaller sunflower group is the possibility that in these tetraploids, there's going to be chromosome leakiness there too, where because they're polyploid, they can lose chromosomes and still be viable. So when we sequence these things, it's possible we'll have, instead of 68 chromosomes, we might have 61. We might have some really, and I think bioinformatically, we've already talked about how we can try to figure that out. It's I think the bioinformatics side is kind of a cool thing, the idea that you can take that sequence and then infer based on read depth of the chromosomes, how many copies of each chromosome are even present. How many of those come from annulus, and how many of those come from tuberosis. So I don't know.
This is super fascinating.
Sorry.
I think we better move to the Land Institute folks, so we have enough time. And we do lose some people at 1, but the meeting goes till 1:30, so there's still plenty of time for their presentation, some discussion.
Good afternoon, everyone. My name is Pheonah Nabukalu. I'm a perennial sorghum breeder with the Land Institute, and I'm based at the University of Georgia, Tifton campus. So today I'll be giving you a few updates on perennial sorghum breeding. So the path to perennial sorghum breeding starts with interspecific hybridization between a sorghum bicolor and a sorghum halepens. So the process is supposed to blend the cultivated traits of Sorghum bicolor with the perennial traits of Sorghum halepens. In the top picture is Sorghum bicolor, that's a cultivated sorghum grown globally. It's 20 chromosomes and it's our source of grain yield and agronomic uniformity. In the lower picture, we have a Sorghum halepens, which is a wild perennial relative of sorghum, also known as Johnson grass, and it's our source of rhizomatous perenniality. So the process of wide hybridization through the process of wide hybridization, we aim to combine the best traits from both species. However, when the two species combine, things become a little bit complicated as a range of ploidy classes emerge. Historically, our breeding program focused on developing tetraploid perennial sorghum, hoping to take advantage of its potential polyploid benefits of high plant vigor and perenniality. But the system turned out to be a little bit complicated, both in the plant phenotype as well as the complicated genetics. Generally, tetraploid plants tend to have excessive plant height, or they're very tall in plant stature. They have excessive tillering and branching, all which complicate management of this crop in the field. Tetraploid plants also tend to have non-synchronous flowering and maturity, which affects harvesting time. Again, the tetraploid plants also produce small brown hulled kernels that affect grain yield and grain quality. But beyond even these physical attributes of tetraploid perennial sorghum, tetraploids generally have a complicated genome or complicated genetics. So they tend to be highly heterozygous with heavy genetic load, and they are very slow to reach homozygosity. So another ploidy that we obtain from crossing Sorghum bicolor and Sorghum halepens is the triploid category, a category that may appear to be less ideal for direct cultivation because these triploid plants tend to be highly rhizomatous, they are highly sterile, and they also tend to have a wild type of phenotype. So I'm putting some bugs on these plants, trying to get some few seeds trying to do some crossing, and at the same time also self this plant. However, despite of all those challenges of triploids, it turns out to be a very important genetic resource in our breeding program. A few times or occasionally, these triploid plants produce very tiny seeds that turn out to be either diploid or tetraploid. And on those rare occasions, some of these diploids turn out to be rhizomatous, perennial, and diploid. So it's these unique qualities that make triploid plants a very important genetic resource in our breeding program. They serve as a genetic bridge, and it's the foundation genetic resource that leads us to rhizomatous diploid perennial sorghum. So currently, our breeding program is focusing on developing diploid perennial sorghum, mainly because of the simpler genetics, and on top of that, the plants tend to have a more crop-like phenotype. So diploid plants tend to have more compact heads with large seeds, and they are easy to thresh. And on top of that, I mentioned the crop-like phenotype. So these diploids are still under evaluation. What we know so far is that they exhibit minimal rhizome development. While they can easily survive in tropical and subtropical environments, we still need to carry out long-term evaluations on their survival under temperate conditions. And another study that we are trying to understand is also the mechanism that's leading to the production of these diploids, because it's not much as they're coming from triploids, the mechanism that's leading to the production of these diploids is still not very clear to us, much as we also think that probably the Sorghum halepens could be producing some monohaploid gametes, pollen gametes that probably mate with the gametes of Sorghum bicolor. That's how we end up getting these diploid perennial sorghums. So this slide is just to contrast between the diploids and the tetraploids. It's just to test the effect of ploidy on developmental and perenniality related traits in perennial sorghum. So starting with a flowering time in the upper right panel, we see that tetraploids generally flower modestly later than the diploids, shifting mean flowering time from 57 to 61 days. So while the difference is not really extreme, it's directionally consistent with the delayed developmental transitions that we see with tetraploid perennial sorghum. Again, looking at the panel, lower right panel, we see that the effect of ploidy on perenniality related traits is much more pronounced. On average, diploids almost produced no rhizomes compared to the tetraploids, with an average of about 19 rhizomes per family. The same pattern was seen with above ground biomass in the upper left panel, where the tetraploids produced more than twice the above ground biomass of the diploids, which probably reflects the high vegetative vigor in the tetraploids compared to the diploids, and also resource allocation to above ground to shoot growth in the tetraploids. Again, looking at regrowth after cold stress, on average, we saw that the diploids exhibited minimal regrowth compared to the tetraploids, or with an average of about maybe 1% compared to the 38% that we saw with the tetraploids. I'm bringing up these results just to help you visualize the journey that we have to travel to make these diploids more robust in terms of perenniality. So this comparison was done earlier on in around 2021 in Tifton, Georgia. With this slide, I'll just situate where we are currently in the breeding trajectory of perennial sorghum. So this slide summarizes more than 80 years of sustained breeding progress in developing diploid perennial sorghum, starting with the interspecific hybridization between cultivated sorghum and Johnson grass that was done in 2017. So we've advanced through successive generations, selecting for winter hardiness in these diploid perennial sorghums to where we are currently at F7 at this current state. So one key feature is that since these diploids do not produce robust rhizomes, we've had to select for winter hardiness directly in spring. For the tetraploids, we could just use rhizome development as an indirect selection measure for winter hardiness. But since these diploids exhibit minimal rhizome development, we have to take the plant through a winter and then select in spring. So right now we are evaluating these diploid perennial sorghums we are evaluating them in multiple locations, including US Midwest, that's in Salina, Kansas. We're also evaluating them in Southeast Tifton, Georgia. We're also evaluating them in the drylands of East Africa, particularly in Uganda. And across these sites, we are selecting these diploid perennial sorghums for grain stability. We are also evaluating them for regrowth, which in the subtropical and temperate environments, it's regrowth after cold exposure. And in the tropics of Uganda, we are evaluating them for regrowth after drought. So across these sites, we are selecting them for all those different phenotypic traits, and building on that foundation, we are also starting to implement genomic selection in our breeding work, integrating multi-environment phenotype data with genotype data with a goal of enhancing or helping us in speeding up selection and also improving the prediction accuracies. So with this work, we are collaborating with University of Makerere and also University of Illinois. So for this slide, I just want to mention or just to highlight that our work in Uganda implements advanced perennial sorghum breeding with farmer engaged or farmer participatory evaluations. We are currently carrying out multi-location trials in 2 agroecologically diverse environments in Uganda. And at these particular sites, we are evaluating these materials for grain stability. We are also evaluating them for tolerance to key biotic and abiotic stresses, and also regrowth after drought. In parallel to that, we are also working directly with farmers in Uganda through on-farm demonstrations and participatory evaluations. And with this work, farmers get to score for traits that matter to them locally. It's very interesting to see that some of the farmer preferred lines also turn out to be among our best performers, in that this the feedback from farmers guides us in our selection indices and also in advancement decisions. So for this work, Uganda serves as a core development partner, where breeding, farmer priorities, and system level impacts all come together to help us build a perennial path for smallholder rain-fed agriculture. So another update from our program, we are deliberately evaluating perennial or sorghum for downstream uses and opportunities. So on top of using a perennial grain, sorghum, as for human consumption, we are also evaluating it as an animal feed. We are currently collaborating with a poultry scientist at University of Georgia, and we've just completed a standardized rooster essay quantifying true metabolizable energy and amino acids of perennial sorghum. Our preliminary results are very encouraging, as we see that perennial sorghum has a true metabolizable energy of about 3,361 kilocalories per kilogram, which is very much comparable to that of corn. Crude protein values are also very high in perennial sorghum. One limiting factor with perennial sorghum is the low lysine levels, which is well characterized, and it's one nutrient that has been routinely managed in commercial feed formulations. So this study or this work supports perennial sorghum as fit to either serve as a partial or a replacement of corn in poultry rations. In parallel to that, we are also waiting for results. In parallel to that, we are also waiting for results from another scientist on forage quality and stover feed value of perennial sorghum. So these analyses help us or guide us in quantifying or in understanding the full grain to forage potential of perennial sorghum, especially for where farmers are intending to use it in their mixed crop livestock systems.
Were there any traits that the Ugandan farmers that were participating in your program, any traits that they desired that surprised you?
Yeah, they're mainly interested in sorghum. I mean, first and foremost, they're interested in tall sorghum varieties or high biomass sorghums, because what happens is that they feed the stover or they feed the biomass to their livestock. So on top of harvesting grain, they would want a crop that is rich in biomass that they can feed whatever remains anyway to their livestock. And the other trait that they are interested in is also threshability and large grain.
Flavor, is that a factor for the farmers?
Which one? Sorry.
Flavor, like the flavor of the grain?
A flavor. No, we've not yet fully started feeding it. I mean, they've not yet started consuming it. So right now we are still should I say demonstration? We are mainly looking at the phenotype. So it's not yet consumed, if I may say, be clear with you. But they're interested in brown grain, seed color.
Quick question. I might have missed it, sorry if I did, but I saw the figures of the tetraploids like doing really well and making a lot of biomass, but what was the reason you didn't want to go with the tetraploids again?
Yes, so the tetraploids, first and foremost, the phenotype, because They tend to have excessive tillers. I mean, they don't really look crop-like. That most times when you present it to farmers, or if anyone sees it, they feel like it's not really should I say, a suitable crop. I mean, it's kind of different from annual or sorghum. And even then, it's a little bit hard to manage. I mean, like they lodge in the field because of the the excessive height. And then they also don't flower at the same time. They produce so many heads at different times, and then all these heads mature at different times. So you can't easily harvest that crop at the same time, because while some heads are mature, others are just coming up.
Do you think there's any way to improve the tetraploids for some of the agronomic traits that you like?
We've tried to improve, but it just takes forever. Like for the diploids, while it would take you maybe about 7 generations to, bring it to uniformity. For the tetraploids, I think you would need about maybe 27 generations for you to, standardize it or see a uniform crop through selfing. So it takes a longer time anyway.
So for perenniality, you said that in the temperate test areas, you were testing regrowth after winter kill or winter dormancy, whereas in Uganda you were testing after drought.
Yes.
Do the same genotypes that do well after winter do well after drought, or are those completely different?
Yes, because the perenniality that we are targeting is mainly coming from rhizomes, and what we've observed with the it's kind of something that you can transfer to it's something that you can borrow from the tetraploids to the diploids. So what we say with the tetraploids, like lines that are more rhizomatous or with a bigger, more robust rhizomes, are likely to overcome winter or survive after winter. And again, those are the same, that's likely to transition to the tropics, like where lines that are more rhizomatous have high chance of surviving a drought. So even for the diploids, lines that are surviving in the subtropics, when they are tested in the tropics, they have high chance, as long as they have a rhizome capacity or the ability to produce rhizomes, they're likely, or they have high chance of surviving drought.
Good afternoon, everybody. Thanks for letting both Pheonah and I come and give a little update today about the crops that we work on. I would like to talk to you today a little bit about Sanfoin and the breeding program that we've had going at the Land Institute for a little while. My name is Schlautman. I have a technician named Spencer Barabal who's done a lot of work for us, and a data scientist named Bo Meyering, who've kind of rounded up the legume team at the Land Institute. We started our breeding program in 2019 and kind of chose this crop as one of the top candidates when thinking about developing a perennial pulse or perennial grain legume crop. It's native to Turkey and the Caucasus Mountain regions of Eurasia. There's an existing industry, mostly in Montana, Wyoming, Idaho, using Sanfoin as a forage crop. And this is an image of a grower we work with in Montana, cutting a field for hay. It's a really fascinating crop. It grows well and is used in these regions because it's adapted to sort of higher pH soils, high elevation. In this particular region of Montana, the growers often have yields that are higher than alfalfa, and that's mostly because they only get 1 to maybe 1.5 cuttings of hay per year, and it'll out yield alfalfa in a sort of single cutting situation, but doesn't have the same regrowth rate and potential. Why Sanfoin? I think there's multiple reasons. This is a picture of a field that is being used for seed production for the forage industry in Montana. You can see lots of little pods sitting here in windrow, and it has fairly high seed potential already. Some of these growers are under irrigation, at least, are hitting 1,000 kilogram per hectare on a regular basis. There's a lot of existing germplasm in the USDA and other germplasm banks around the world. And there's a couple other breeders. That's maybe a little different than finding other sort of domestication opportunities for other perennial pulses. It's already a high quality, valuable forage, and those of you who've been in the perennial grain space for a while have kind of heard about the sort of dual purpose opportunities that we might want to be developing for producers, and it's currently already adapted to the major pulse producing regions of North America. So I don't know how many of you are breeders. I thought I'd give a little background here of sort of how we're thinking about domestication and how it kind of fits into breeding in general. This is a breeding equation. It's a really simple tool that we use sort of to monitor our progress, our metric of success when we do breeding. This delta G here is called genetic gain, and it's sort of a measure of the changes that we see in response to selection on an annual basis. And there's a really nice simple equation that lets us calculate this metric. And so when we think about, how do we improve our genetic gain? How do we get closer towards our goal? In this case, maybe it's seed yield, realized seed yield per hectare of Sanfoin. There's some really important things to do early on in the domestication process, and one of those is just to figure out how to start with a good base population. What are you gonna? What's your starting point that you're gonna conduct breeding long into the future? So if you think about, I guess, our program at the Land Institute, a lot of what we did the first few years was just evaluating existing germplasm and trying to select, not by performing crosses and reproduction ourselves, but just evaluating accessions and choosing right. And you can kind of imagine here your y-intercept is going to be kind of your starting point in terms of what your yield potential is in existing germplasm. And we had a lot of fun doing this, and we have work ongoing evaluating the many different species in the Onobricus genus, the many different accessions available within the Onobricus vitiifolia Sanfoin species that we currently work on with multiple collaborators and students that have now received their PhDs from Turkey. And you can see there's many different flower colors and pod shapes and sizes in the genus. If we think back to our breeder's equation, this year in Montana, in some of our trials, we averaged around 520 kilograms per hectare of seed yield. That's with the pods removed. It's a single seeded pod. Over the last few cycles, it seems like we're achieving around a 19 kilogram per hectare increase in yield. And so you think about, well, what is breeding and where are we shooting for? We're not really supposed to project out like that, but you can think of, in a career, 30 years left of my career as a breeder, where might we end up? And at this current rate, it's somewhere a little over 1,000 kilograms per hectare. And that's pretty interesting to me. That's roughly what the dryland lentil yields are in the region right now. It's kind of exciting to imagine, well, maybe it's possible to get there. But getting there really depends on the way that you're doing the breeding and the way that you're measuring and the patience that you have. How fast do you want to go? So you can kind of see this in this figure here. Once you have selected your base germplasm and you start, ongoing cycles of selection, which is kind of represented here in 2020, assuming nothing really changes and everything's the same for the next 30 years, you have a sort of consistent slope. This is your genetic gain and the rate of progress you'll have. This is a picture of some of our breeding plots in Montana. Spencer here is in the left side of the picture. These are half sib progeny plots that we use to evaluate germplasm. And then here's Sean again, the farmer you saw earlier on. And so some of the ways that we think about, maintaining a certain rate of genetic gain is kind of developing tools that we think can last in our program for a long time, coming up with image processing methods to consistently measure seed size, shape, classifying seeds that are coming out of the combine as cracked or depotted already. And Bo's done a really nice job in our program developing these sort of web applications that we can use. So we're doing some seed image analysis. We're working on developing more machine learning algorithms to understand establishment and weed pressure and other characteristics about plots. That's been kind of Bo's key work the last couple of years. when we think about, what else can we do to improve our rate of genetic gain, some things that can happen, especially during domestication, is that we have opportunities to maybe maintain the same rate of genetic gain, but quickly sort of jump up to a new y-intercept. And that might happen by identifying new interesting traits, domestication traits, stopping, shattering, things like that. And while we do most of our selection right now in half-SIB progeny plots, we still evaluate some individual plants in the field, and we've been discovering some unique phenotypes that we're trying to develop populations out of. And this can look like, for example, a pod that has 2 seeds. Typically, you can see in the background, we have inflorescence that's not branched at all. And we've been finding more and more individuals in our populations that have these sort of branched inflorescences that may be good or may not, similar to the sunflower discussion we had early on. But finding one of these traits is potentially one of the fastest ways to slide up y-intercept and keep going in terms of breeding. The other methods that are available to us, many of you probably know, we can do things like genomic selection, which potentially allow us to make more selections, one selection per year, but compared to maybe a selection cycle every 3 years in a half-SIB progeny evaluation approach. And you can think about, not only are you doing genomic selection, but even within a genomic selection framework, you can potentially change your slope or your rate of genetic gain by, shifting adding more plants that you're genotyping and evaluating in the process. So one of the things that I don't know if Lee or anyone from the Land Institute's presented on in the last year or two that we're looking at the Land Institute is thinking about another approach to change our rate of gain that's potentially more accessible not only to our programs at the Land Institute, but programs internationally, and that's called phenomic selection, which is in many ways a similar concept where we're estimating sort of additive relationships among plants in our breeding programs, but instead of using DNA and sequence data, we're evaluating the use of sort of NIR-based scans of seedlings to estimate those relationships and make the same predictions. And so you can imagine that perhaps the rates of genetic gain that are possible are not the same as the potential for genomic selection, but perhaps the cost per unit of gain could be quite a bit less. Our crop stewardship teams recently kind of helped us come up with a name for this crop, and they're calling it Perennial Baki bean right now. And Baki is a word derived from Turkish that means eternal, everlasting, or remaining, and it kind of represents both the region where Sanfoin's native to, its center of origin, and also our ongoing collaboration with Turkish colleagues. This is kind of what the crop looks like. You can see after the pods are removed, here's a couple pods up here yet. It has a sort of a brown hard seed coat that can be split just like lentils, and the inside of the split Sanfoin, I guess, is this really beautiful yellow color. We've had people that are experimenting with different uses for the crop, using it in salads and soups, making hummus out of it, milling it into flour and using it in different gluten-free applications. And we've also had a postdoc in our lab. His name was Evan Crane, and he's done a lot of really good things the last 4 years, and recently became a lead scientist in the crop stewardship program at the Land Institute, focusing on sort of bridging the food science and breeding plant physiology efforts. And so this is some of the results of work that he was a part of when he was a postdoc in our program. We learned some basic information about the nutritional attributes of the species. It's got really high protein content, similar to soybean, an interesting amount of crude fat compared to other pulse crops, not nearly the amount of soybean, but so we've looked at some basic information about the nutritional attributes, and then we've done some mice feeding studies and other screens looking for anti-nutritional compounds that we have not identified any in particular at this point. We're still working on trying to achieve the generally regarded as safe status for Sanfoin grain and ingredient forms in the U.S. We've got a ways to go in terms of the types of research that they'd like to see results from. But yeah, that's one of the key hurdles that the crop needs to move through yet to become something that works commercially in North America. So,
Hey Brandon, do you see a decrease in yield over time with this plant, like over subsequent years of growth? Have you guys looked at the effect at all?
we haven't done a lot of great research on looking at yield stability, but what is exciting is that we know there are fields of Sanfoin that seed producers here have that are 16, 17 years old. We went to a field last summer that was 40s, planted in the 1970s, over 40 years old. And so under the right environments, we know that it can survive for a long time, just like Kernza and Silphium and the other crops that we're working on. But obviously, seed yield potential is likely to go down seemingly over time. And potentially, if you look at Kernza in environments like Kansas or the western part of the U.S., can still achieve fairly high yields after certain environmental events. A drought that's followed by a really nice wet year seems like it. We can have really good intermediate wheatgrass seed yields still 5, 6, 7 years after establishment. So, yeah, something that we have to learn about.
Does it taste like lentils? Like that?
It has a very doesn't have really a strong, memorable flavor, which I think is actually a really nice quality. Most of the other pulse candidates that we tasted had memorable flavors that maybe weren't something that we wanted to eat on a regular basis. So I think we got pretty lucky choosing this candidate in terms of the lack of anti-nutritional compounds and weird flavors. Most of the other things that we've tested in many legume crops in general have quite a few compounds that they have in the seeds to protect the protein they're packing there.
I have a kind of a challenging devil's advocate type question. So Brandon, I love the chart that you put up, thinking through what it looks like to domesticate and then improve a novel crop. And completely take your logic around the possibility of changing the y-axis through finding key domestication traits. We've seen that happen in pretty real time with pennycress, and it's well documented. But then when you continue those lines out of the future and you get to yields that are like double what we have in lentils, I start to get skeptical. And, if that's possible in Baki bean or Sanfoin, what makes it fundamentally different than lentils, which has received significantly more breeding input and has topped out, so to speak, or probably is at the stage that we're at with wheat and oats and lots of crops where, breeders are able to make pretty modest incremental gains. And it's a fully domesticated species. So it kind of implies that there's inherently could be a much larger yield potential. And it's very possible that the, conventional breeders out there working on lentils just aren't pushing the envelope as much as we tend to in this perennial grain community. And, maybe there are breakthroughs that are possible, but how do you square those two notions that this has kind of been tried in somewhat related species that do tend to, plateau or at least reach a level of pretty incremental yield gains, and yet we're holding out the promise of, breaking through some of those same plateaus?
There's a lot of potential answers to that question, but I think one of the unique things about both Sanfoin and intermediate wheatgrass is that they're outcrossing highly heterozygous species, a lot of diversity that's there right now. And I think there's this other reality that maybe we don't feel like we've seen the yield gains in lentils and some of those species, but you can look at corn and soybeans in the US, and we've 10x'd them over the last 100 years of breeding, and we still see, realized at a sort of whole United States scale, half bushel to a bushel gains in yield, genetic gains in yield in crops like soybean that are, selfing species. And so it's maybe a cost of genetic gain, and what's the level of commercial concentration of capital that's possible based on the supply chain available there. That could be one of the determining factors in wheat. Maybe it doesn't seem like that's true, but yeah, I think it's a to be determined thing at this point. So
Yeah, I'm not trying to take down the thought process. I'm trying to understand and think through how we can all, as a perennial grains community, project out into a very uncertain future. It's uncertain for many reasons, including what's going on with the genetic potential of the species. And so, yeah, that's helpful. Thanks, Brandon.
Yeah, I think my goal really of showing that is to try to understand what is the domestication that we're doing, the breeding look like right now. Maybe not where we're going to get to, but how there are different ways to increase genetic gain. For example, my program at the Institute, maybe I can afford to evaluate 3,000 lines a year and genotype 500. That's a small number. Imagine, we don't change anything in terms of the allocation to individual programs, but if you scale to 10, 20 programs around the world, there's a lot more evaluation happening. There's ways that you can spend more, genotype more, and those are the hard questions that I think a lot of us in this domestication space are facing, which is how much money do we allocate towards any one of these crops in terms of genetic improvement, and how do we judge the success and metrics of sort of crops at this very early stage when a lot of them were just establishing for the first time real breeding programs at all. There's a lot, I was talking at the Land Institute the last week with some colleagues, and in a lot of ways we're just barely doing breeding, right? Some of the crops we only have a single environment, and we really can't even estimate heritability because we don't estimate G by E interactions for a lot of the stuff. So we're very early on, I think, and in many of these species. So I think the opportunities are high.
For the polyploid presentations, they're in canola, they're working on trying to get apomixis working for hybrid stuff. And I'm wondering if there are any kind of species that are in the family that are apomix for either sorghum or sunflower.
Sunflower, I'm not aware of that, but I can check into it. Yeah, that's interesting. That's an interesting idea for sure. I think in the case of what we're working on, the assumption is we want to use clonal propagation anyway, which is as good as apomixis.
But it might help with fertility though of the seed.
Yeah,
not necessarily just like planting.
That's true.
I can comment that we've not really tried to search out for apomixis in perennial sorghum, much as I know it's in annual sorghum. There's a genetist, there's a cytogenetist here who is looking at apomixis in sorghum, but we've not tried searching for it in perennial sorghum. That possibility is there, but we've just not yet explored it.
Matthew, do you want to give a nutshell for folks who might not be as familiar with what that is?
Oh yeah, it's just asexual propagation of seed or making of seed through an asexual means. Yeah, so if you have a triploid, for example, you can still get good fertility from a triploid, seed fertility from a triploid through apomixis.